Hi, everyone. On behalf of Metabolon, I'd like to welcome you to Driving COVID-19 Research Forward. My name is Carolyn, and I'm the host and moderator for today's event. Before I introduce our presenters, I'd like to provide you with some brief instructions. The presentation has been muted, but we welcome questions that can be answered during the Q&A session at the end. To submit your questions, please go to the Q&A button at the bottom of your screen. Now I'd like to introduce our presenters for today. They are Dr. James Heath, President and Professor at Institute for Systems Biology, and Patricia Sheridan, PhD, Senior Study Director in Discovery and Translational Sciences at Metabolon. Welcome to you both. Patty, the presenter role is yours.
Okay, thank you so much. And again, Jim, thank you so much for taking time today to talk to us about this research. And really, this is going to be more casual question and answer about the research that you're doing at ISB to look at COVID-19. So if you would like to tell us, to start off by telling us about the study that you are working on, and then we'll go from there.
Sure. So we launched a study on COVID-19 patients about, well, more than 2 months ago, starting around late March, with the ultimate goal of consenting about 200 patients. So this study has many parts. So we're at about we're not quite at 100 patients now, but we're almost there. So on each of these patients, we're taking blood from them at a point where they get diagnosed, and then at a few points after that, about a week after, a month after, and then ultimately we'll be doing longer term follow-up. And we are analyzing that blood for many things. We're doing a lot of single cell analyses, try to understand what are the protective immune responses that can be developed against the virus, as well as what are maybe non-protective immune responses. What's the heart of immune system dysfunction in the most severe patients? We're looking at broad surveys of metabolites, as you guys know, and proteins. We're doing whole genome. And we've also been doing autopsies on patients who have succumbed to the disease to try to understand the, sort of the extent of the disease, and I guess in its worst cases, and the various organs that the virus infects. So a big study. We've, let's say we've probably collected 150 blood draws so far on approaching 100 patients. And so we've developed, it's already a very big data set that we've developed. And we're just starting to begin writing things up and sending them out. I think we have one paper that we sent out, but there are several more that are in the works.
Are the subjects that are in the study, are they all hospitalized, or are these across the spectrum of just, that they were diagnosed but may have a full recovery, or are we looking at a spectrum of severity?
Yeah, so the goal is to look at the whole spectrum of severities, from basically asymptomatic to ICU patients. The practicalities of it have been that by and large, for this first 100 patients, most of them are in hospitals. So they're either, pretty ill or severely ill, either ICU or just in the hospital. To do the, more moderate infections, we had to get mobile phlebotomy going, and we have that going now. And so we're beginning to look at patients who are being treated by telemedicine or convalescent patients, et cetera. So right now, I would say the picture that we have of the disease is one that affects, for the most severely affected patients. We don't really have a deep comparison point yet of mild infections and symptomatic infections.
We have a question from one of the participants that's wondering, what aspects of COVID-19 is metabolomics particularly suited to address, and why it was included in this study?
Oh, well, I think, pretty much, I think a lot of us who are doing COVID research right now were transplanted from some other field. Um, but there's almost no disease one can think of that you can't at least view as a metabolic disease, whether it's cancer or diabetes or probably COVID. It depends on, there's this adage of, looking for your keys under the street lamp because that's where the light is. And so we tend to be a little bit married to sequencing methods as a way to, look at disease states. But if you go and interrogate any disease at a deep level using metabolomics, then you find that there are an equal number of metabolic susceptibilities and metabolic processes that are dysregulated. It can be a little more challenging, although with the tools, for example, the Metabolon, you get on the order of 1,000 metabolites from to these patients. It can also be remarkably revealing.
So you brought up that, it makes me think about the comorbidities that seem to be associated with greater severity, things like diabetes and obesity, high blood pressure. Because these are hospitalized patients, you will have captured all of that information?
That's right. That's right. So, I think if from the outside looking in, doing a study like this, especially when the preparation time for the study was about 3 days. And so pulling data out of the EHR, or there's also another type of data you pull out called REDCap that you get basically when you're taking blood out of the patients. So looking at comorbidities, at different drugs these patients are taking, at things like body mass index, et cetera. All of those are put. So let me give you a picture of how we envision the study evolving. And so you have a lot of clinical measurements that range from ferritin to blood pressure to et cetera. We also, on these patients, are doing very in-depth single-cell analyses. So with these single-cell analyses, which are pretty crazy expensive, but they're very high information content, one can begin resolving, even in this sort of Wild West scenario of how drugs are being used right now for treating COVID-19 patients, one can begin resolving how those drugs work and what their effect is. You simply can't do that on every patient. And so what you would like to be able to do is to take those inferences that you get out of these very deep analyses and then begin looking at how they are reflected in things like the serum metabolites and the serum proteins, and ultimately look and see how they're reflected in the standard clinical labs that are done. Maybe the labs aren't going to be 100% effective, and you might have to add a few things to really resolve the effect of some of these therapies. But that's sort of how the strategy is going. So with the very in-depth single-cell analyses, we have pretty impressive data sets. We can tell from just looking at the blood of a patient, if the patient's in the ICU, we can tell they've been treated with a couple of different types of drugs. We can, to some level, anticipate what the outcome will be. And now we've just now started getting enough samples that we can do the sort of bulk assays, the kind of things that Metabolon is doing with us. And so we hope to have that data back within the next week or so that we'll be able to now do simpler assays to look at these types of outcomes and effects of drugs.
Ideally, these data in combination could be used to identify people early on who may be more at risk for more severe consequences from COVID.
Yeah, that's a great point. And I don't know if we know yet how to do that. I'm pretty sure we don't. There's been a lot of comorbidities that one thought would actually correlate strongly with disease severity. And there's a really beautiful paper put out. It's on bioRxiv or medRxiv. You can go find it. Just came out 3 days ago from John Wary at University of Pennsylvania, that find that there's not actually a very strong correlation with a lot of these comorbidities. There probably are with some, but
Right.
not all the ones that you would think of. But something that we're finding that I'm sure is going to be reflected in the metabolic assays is, so the patients who are most severely affected. So normally you have a sort of a symphony on how your immune system should be responding to the virus. You have your sort of first responders, which are neutrophils and things like that. Then you have T cells, which help educate B cells, and then ultimately you get antibodies. And in the most severely infected patients, basically the T cells are unable to mature. There's no immune response that evolves out of that cell population, which basically stops this whole symphony in its tracks. And that is absolutely going to be reflected in other more broad serum metrics like the metabolome or the proteome.
Excellent. We have some questions we have additional questions from the audience. This is kind of a forward-looking question for your project. Someone's interested in if you are looking to include young people with apparent Kawasaki disease in the metabolomics study, given that they're seeing the new consequences that there appears to be a Kawasaki-like disease that's associated with coronavirus in the young.
Yeah, so there's a national strategy for doing this kind of thing. So, everybody launched studies very quickly that was trying to, when, 2 months ago, even now it's still happening, people are launching studies. And if you want to get a study launched quickly in IRB approval, you don't study kids. It just takes, it basically 18 plus is what you put in. But now, a couple months later, there are some of these studies that are now beginning to include kids, including there's one here in Seattle at Seattle Children's Hospital. There's some on the East Coast. I know Stanford is looking at whole families. So the NIAID, Fauci's organization, is doing a nationwide 1,000-patient study that will include all these cohorts. Our part of this study.
Yes.
And so we will be able to start looking at kids, at non-asymptomatic people, at a host of things. It's really a game of numbers to really get the statistics up, to try to resolve things like the pediatric conditions that were just referred to. And yeah, we're not actively recruiting those patients, but they will be incorporated into our databases as this national study matures.
Right, and we're excited here at Metabolon. We're actually a part of that study, and we'll be running a lot.
You are, that is right.
So yeah, we do expect to get some really powerful data out of those as that study expands. But you bring up some really interesting points about how clinical studies in response to an immediate event are designed and how you have to deal with kind of the limitations of what the IRB will allow in those kind of first studies. We have some additional questions related to, I think we hit upon ideas of, predicting severity necessarily. And then I guess you spoke a little bit about responders and non-responders. Are you intending to work on specific treatment trials? I know in this you will be collecting data of the treatments that the patients are on, but will you be prospectively enrolling for treatment trials of various antivirals or other medications?
Yeah, we are. So right now we're looking at drugs that are already in the clinic. There's a bunch of suspects that pop up in the news occasionally. Remdesivir, which recently got approved. There's tocilizumab, which is an anti-IL-6 that comes out of Roche, I think. And then there's convalescent serum, of which the very, very first reports are beginning to come out. There's going to be antibodies. There's going to be a host of different vaccines. And then there's a number of immune modulator drugs that are being tried. And so we're in conversations with some of these different companies to try to, and we will be using the sort of deep analytics that we do to understand the effect of those drugs. And, you know, a lot of this, so let's look and see how the drugs have been used so far, because it's really been, it really has been a Wild West.
Right.
So as I think, not everyone's aware of this, but if there is a drug that is approved for some indication, it can be prescribed by a physician for, as a physician discretion, if it's in what's called the pharmacopoeia. And so when we, in the first, say, 10 patients we looked at, we had remdesivir, we had chloroquine, we had antibiotics, we had tocilizumab, we had vitamin C. I mean, it's just a whole range of things, and they're all being used in according to physician discretion, which means that every physician treating patients might be using them slightly differently. And you know, with the very deep analyses we're doing on these patients, we actually can still resolve at least some of the effects of these drugs. With the, you know, now life is getting to be a little more normal. There's some actual double-blind clinical trials, which is really what you have to do to understand if a drug works or not. And so we will be involved in doing some of these. I think it's still fair to say that this is a disease for which there is maybe not no treatment, but there's minimal treatment. You know, remdesivir does seem to have an effect. Convalescent plasma has an effect on at least a subset of the patients, but not all patients. And, you know, that'll change over the next few months as these double-blind trials begin to resolve not just what drugs to use, but when to use them. There's a real art in how these drugs are applied in patients as their condition worsens or improves. You know, what the timing of these drugs. It's not like you just give someone a drug and wait for the conditions to get better. You have to, sometimes you have to wait till you think there's going to be a spike or a cytokine storm or something like that before you would give some of these immune modulators.
You alluded to this, you know, you are using a variety of techniques. You talked about the different single cell analyses, proteomics, you know, metabolomics. Are there statistical tools or model building that your group will be doing to integrate these various?
Oh gosh, there's crazy numbers of that. So one of the goals we have here is to be able to identify what immune responses are protective. And there's some evidence that some of the immune responses that one can have basically just wear out the immune system, at least in the more severely infected patients. So in these analyses we're doing, we're able to get, you know, in single cells, the entire transcriptome, 200 proteins, the T cell receptors, the B cell receptors, all from the same single cells that are floating around in your blood. We can also, in a separate assay that we developed here at ISB, identify exactly which pieces of the virus your immune cells are seeing, what they're reacting against. And it's going to be different for different people. And so what that allows us to do is, in this huge database, follow exactly what those anti-COVID T cells and B cells are doing. Are they behaving as you would normally expect an immune response to behave? Are they basically becoming dysfunctional? To really understand what's protective and what's just noisy in the immune response. And that is, in many ways, at the heart of how one thinks about designing a vaccine. Because you want to have a vaccine that promotes those immunoprotective responses as opposed to just exhausting your immune system, which, you know, is actually quite possible if you don't fully understand the nature of the virus-immune interaction.
Somewhat related to that, I know for now you're taking peripheral samples like blood. If you're taking primary site of infection, are you also intending to do viral sequencing? And you mentioned the autopsy samples that you have. Will you be doing sequencing of recoverable virus there?
Yeah, so we have, we've been doing a number of virus sequencing. So there's a lot of work that's done on viral sequencing out of things like nasal swabs, or these nasal pharyngeal swabs, which patients absolutely hate, and I can't blame them. It turns out you can also piece together the viral genome out of the blood. It's fragmented, but it is there. Okay. And we've been doing that. And that may ultimately provide a better measurement of viral load. It's not the kind of thing you want to do on every single patient because it's a bit pricey. You know, you need like a 50% test per patient if you want to do something that's effective on broad populations. But it's a way to really understand two things. One, is your drug affecting viral load? And two, if you have patients that recur, which you definitely do have patients that recur, is it the same virus, or is it a variant of that virus? And so we are doing that. We're also doing deep analyses of the sites of infection. So in these autopsy studies, we've done on one patient recently, we did just a very broad survey initially to find out where is the virus in this patient. And people think of COVID-19 as a respiratory disease. And by and large it is, but it turns out the virus can almost go anywhere it wants to. And especially in the most severely ill patients, you see it in the kidneys, you see it in the heart, you see it in the GI tract, you'll even see it in the brain and the liver as well. And then you can look at specifically those sites of infection in, say, the GI tract or the lungs or the kidneys. And modern tools of biology are amazing. You can take just a particular little tiny section of the diseased tissue that maybe is 10 cells big and do the whole, do the transcriptome or do 200 proteins out of that region. We've been doing that work with a local company called NanoString. It's remarkably revealing.
Going back to the immune system exhaustion, is that the kind of thing that you think will be reflected in the peripheral metabolomics?
Oh yeah, yeah, for sure. So if you look at, well, let me give you an example of how metabolomics and of how is reflected in COVID-19. So, you know, one imaging modality, so there's a lot of imaging, we're used to seeing lung CTs and things like that associated with COVID. You see this diffuse, you know, mass in the lungs. That's really is what makes people think that COVID-19 is a respiratory disease. But another imaging modality that is more commonly used in cancer and in neurodegenerative diseases is positron emission tomography, PET. And its main value in cancer is that you take glucose, perhaps one of the most important metabolites, if not the most important metabolite that one has, and you radio label it, and you're able to look at where the hotspots of glucose consumption are, and that's typically the growing tumor. Well, one of my colleagues at UCLA, Johannes Saarinen, looked at a handful of COVID-19 patients using FDG PET, not because he was trying to image the virus, but because he had cancer patients that happened to be infected with the virus in early days, and so we got the images. And you see a very significant PET signal. There's a very elevated glucose metabolism in the regions where there's infection. And so that alone, that's a you know, a picture's worth 1,000 words. That picture tells you there's going to be a massive metabolic signature associated with this disease and probably with the severity of the disease. I think if we talk again in 6 weeks, there's a good idea that there's a good chance that we'll have much better resolution of what that picture is.
That might be fun. We might have to do that. This first metabolic analysis done. We have a question. Do you know if the virus attacks or accumulates in the salivary glands, and if saliva collection for testing is possible? And I think we've seen a lot of recent papers that have suggested that this may be a method that could be used that's less invasive.
Yeah. So there is, in fact, evidence that saliva or sputum, which if you had asked me a week ago, I'd have said those sound like the same thing, but they're not.
We learn something new every day.
Yeah. So saliva, just out of your salivary glands, is a place where you can find virus. It doesn't mean that it's going to be a positive test if you have the virus. It may be a little, not such a great diagnostic test. Sputum, in which you're basically trying to cough up saliva from the lungs, is actually a pretty good test. In fact, it seems like it might even be better than these nasal pharyngeal swabs.
Okay.
The challenge is that this is a disease that tends to dry people out. And so it's actually very hard to get a sputum sample. So we may be stuck with nasal pharyngeal swabs unless there's some way that one can get a sputum sample a little easier than just asking people to cough up some sputum from the lungs. But the hope is that, maybe it's a sensitivity issue or what have you, but, if we're trying to get people back to work, antibody tests are going to be the dominant tests that let people back in the door.
Mm-hmm.
You're going to want to know if someone has a fever, if they need to quarantine or something. And you need, rapid tests to be able to do that on a scale. And, it's possible these saliva tests may evolve into that. It's not clear yet.
Well, I know that you have, it's now 11:30. And thank you so much for taking the time to join us. This was really very interesting, really informative. And I do hope that we get the chance in the not too distant future to come back and discuss some of the updates in terms of the data and what your group is finding.
It was my pleasure.
Great. Well, thank you so much. And thank you to everybody that participated and had such great questions.
And thank you. It's a pleasure working with you guys on this project as well.
Well, we're thrilled to be working with you too. It's such important research to be doing.
All right. Y'all take care.
You too. Thanks.